was give us a quick introduction and then tell us about the objectives of this dive, if you would.
Sure. So we're diving on a topped seamount, also called a GEO, and this will be a GEO. It's located within the U.S. exclusive economic zone just to the east of the Mariana Trench. These GEOs are of interest to us because they are very, very close to some locations where the International Seabed Authority is granting leases for future deep-sea mining. These geodes tend to have a very significant crusts that have built up over the rocks. They're called manganese crusts that are filled with all types of minerals and are commercially valuable. And so what we're trying to do is study some of these geodes and try to understand what type of animals live on these types of seamounts in preparation for maybe deep-sea mining in another 50 or 100 years out in this area of the Pacific. We want to get a head start on understanding the consequences of this activity. So that's why we're here. We're going to be diving at around 2,000 meters because we have found last year that there are some magnificent communities of deepwater sponges and corals that live at 2,000 meters depth down here. And yesterday we saw some beautiful corals on one of these geodes, the first one that we did, Pigafetta geode. We're hoping to see the same thing here, and that would be fantastic because then we hope to inform people that these aren't just sort of lifeless, mineral-rich plateaus down in the deep sea, but are in fact home to some really, really interesting animals, and there should be a cautious approach to any type of activities that occurs on these places in the future. The specific place that we're diving on is a ridge that's located on the summit, and it's the type of place where corals and sponges like to hang out because they get good current flow. So we're looking forward and we're hoping to see some animals here.
Thank you very much, Chris. So we've just got the bottom in sight right now. No. You're not on. No.
Hey, Nav, what's our time off bottom?
Watch leads. Right now, I still have 16:30.
Okay.
Thanks.
Wait.
Really?
Is 16:30
50
at 50 meters?
Yes, that's
Oh,
right.
no. Okay.
Okay.
Oh, you're talking
Yeah,
time
time off bottom.
Yeah.
Oh, sorry. Standby.
So I was like, wow, that's late.
No, yeah, I was like, something's wrong there. Well, from a geology standpoint, this looks a little more promising than yesterday.
Yeah, what do you have to your starboard? Yeah, just a little bit. Okay. Yeah, go ahead.
So we have 2 hours, 51 minutes.
Wow. Okay, thank you. We got it.
I'll come down a little bit with you.
Did you hear that?
Yeah.
Does this look steeper than bathymetry had? You guys? That's local does here. Yeah.
I know, I'm very excited.
So we're going to grab a rock straight away? Is that what's happening?
I believe we are going to try to grab a rock straight away, assuming that we can find anything that's loose enough to take.
Okay.
Actually, Chris, do you want to discuss the sampling scheme that we talked about? Or do we want to play it a little bit more by ear?
Yeah, sure.
Okay.
So because we have a shortened dive today, we decided that we should probably collect only one rock and maybe one bio specimen here, just so we can cover a little bit of ground and see if we can get into some of these communities.
Yeah, I think it was 2:30.
So what we're going to do right off the bat here is find a rock, hopefully, and get that locked in and get it in the basket. And so I see plenty of candidates, and I don't know how loose they are, but they look pretty good. And I'm going to let you, Deb, and Patty Fryer and other geologists figure out if there's one you want.
Right, perfect, thank you.
Okay. Yeah, I'd be happy in 260 because
Bridge, Nav.
So, Pilot, can you just let us know when you're set up?
We'll do that.
Thank
Yep.
you.
No problem.
Bridge, nav.
Are you calling us?
No, that was the bridge.
No, it was the flashing pilot sign.
Oh.
No.
But no.
Bridge, be advised the vehicles are on the bottom. Please make your bailout due east. 090. Copy that. Thank you, Bridge.
Watch these. I key you and unkey you all the time, so I wouldn't pay attention to the lights.
Okay.
So I heard talk of sampling.
Yes.
Maybe I'll give these a poke with my skid here and see if they're loose. Does that sound like an okay?
That would be perfect. Yeah, it looked mostly pretty in place, but if we could find one that was loose and just grab it quickly, that would be great.
Sure. Oh, there's a coral on the one I was going to try to nudge.
Yeah, this local zone is pretty steep. My sonar cannot see over it.
Yeah.
Hey, Nav, this is Watch Lead.
Very solid.
Go ahead, Watch Lead.
Is our depth off by like 100 meters?
The depth on the target or the depth that
the depth
vehicles?
from like the high pack to where we really are.
Well, I mean, where we are right now is 2267.
Yeah.
And
Yeah, they don't match up.
Okay. Just making sure. I was a little confused.
Navigator, do you know the depth of waypoint 2? Then, I don't know, maybe compare that to the, just a thought, but
it's, I don't have it anywhere.
might be in the text file if it's still open.
Okay, I'm going to record a couple things real quick and then I'll get back to you, Copilot.
Yeah,
Yeah,
don't worry about me.
we're stable here, so you're fine now. Well, I gave that a nudge, Watch Lead, and they look pretty cemented.
Yeah,
Um,
I believe that.
But something like, some sediment around it down here, if you see anything loose,
What about the one that's in the bottom center right now?
bottom, center,
Just below that big boulder.
you want to throw lasers on for me? Use that as a reference. Can you see lasers, Watch
Yep,
Lead?
that one.
This guy here?
Do you think that might be loose?
I don't know.
Yeah, it's hard to tell.
We can give it a nudge. Maybe. That one's not loose. I think it's not any different.
Yeah. As we get closer to it, it looks less promising.
Yeah.
Yeah, there's the one sort of there's actually two right now in frame that look like they might be possible. One
Right.
to the right and a little bit below the lasers.
Yeah, I see that guy.
And then one that's almost under your left skid or the left box, it looks like.
Okay.
But it's hard to tell from here.
Sure. Yeah. Can't see any of these moving.
Sizable sponge there.
Can you get my mini Zeus and my pilot cam just pointed down at my toes, in between my toes, please?
Sure can.
Thank you. Well. None of those are loose. I mean, we can give it a shot with the arm if you want, with the one on lasers on it now.
Do you think it has a possibility of being loose?
I don't know. It's just so hard to tell.
Yeah, it is really. This is not easy to pick one that looks like it's not in situ.
Yeah.
We can give it a try, and
Co-pilot
if it doesn't work, we're just going to keep moving, I
Go
think.
ahead, Nav.
Okay, you want to give it a shot?
Yeah.
So there's
Okay.
2 waypoint files. I assume we're using the new dive track one.
Okay.
And in that one, waypoint 1 is 2261 meters. And waypoint 2 is 2200 even.
Okay.
Can you bring my mini zoos way right? Check out the crabs, please.
Yeah. Thank you, Navigator.
Yeah, so it looks like they're both far off from what we see on the contours at least.
Yeah, this might be 100-meter bathymetry or something.
Okay, let's just give it a shot here. You want to spin me out?
The dog's ready.
If we do grab one, we're going to go starboard, I believe.
Totally fine with me.
Okay.
Copy that.
Well, actually, that might be a little trickier where we are. Can you bring port wing in? Yeah, that's probably good right there.
I can bring it in further, but
Yeah, go ahead. That's probably good, yeah. Maybe we'll go port, actually.
Cool.
Belay that last watch lead. We're going to see what's easier.
Okay.
you want me to follow you with mini zoos?
Yeah, please. So I want to make sure I don't get that rock, that big feature to the right over there.
Yep.
Yeah, so.
Going down with mini zoos.
Yeah.
Come on, loose rock. I don't think they're actually all in place. I just think they might be fairly well cemented in.
Yeah.
Yeah. I mean, I could be wrong, but there should be some that are not in place. But this is not one of them.
Yeah. Unfortunately, this guy seems pretty in place.
Okay. While you've got the arm out, can you just tap a few other things? And
But he's not in place anymore.
You think you can get him now?
Yeah, I
Okay.
think so.
I turned it off.
Looks like that.
Can you come down like
Deb,
bottom left?
it is.
The port here.
I'm sorry, Patty?
Mini zoos.
Coming down.
I said it looked like that boat moved. Did it move?
Yeah, it did move. We didn't think it did, but
Should be
Then
good right
they-
there, I think.
put a little more power into it.
Perfect. Come back up to it. Stand by.
Excellent. See, there are tons of loose rocks. Pilot, if you wouldn't mind giving it a spin.
Sure.
So it's great you loosened up a few others for the next ROV here.
Now go
Very
ahead.
true, Chris.
Want to get it back in the light?
We should remember this as a dredging spot, Deb.
Good.
Deb.
Just missed it. coral cutters there. That's good.
I'm glad we're making you guys so happy today.
I was nervous about that. It was cemented.
It was just in a shallow
We're trying
bath
to
of
figure
sediment.
that out ourselves.
Figure what out?
I think it's because even with a little bit of a problem getting in the water, the ROV is down and diving, and that's making us quite happy.
Good, good. Yep.
Good?
Yep.
Okay.
Port.
Come
Is that
on.
where you're going?
Port.
Yep. Thank you.
Sure. Come down.
Nice.
Awesome. Thank you so much.
Sure. I'm gonna come outboard with that, see if it just closes.
Yeah.
So it's a little easier. There she goes. Thanks.
No problem.
Is that all the way outboard?
I think so.
Give it a little,
A little coaxing.
yeah.
Nav, do you have a moment?
Come left.
Go ahead, Watchley.
So I think the plan for today is sort of a, I don't want to say slow and steady, sort of a moderate and steady pace as we try to make as much way along the track as we can in 3 hours.
Down there.
Okay.
I think we're going to be fairly, we're going to try not to be stopping all the time.
Copy that. We'll put a move in as soon as we're completed the sampling.
Great, thank you.
Yep.
Okay. Hydraulics off. You made a mess, huh?
I'll center up your mini Zeus real quick.
Yeah, thank you. Yeah, you just give it back to me whenever you're done, that's fine.
Yep.
So that looks like the local high co-pilot. Pretty much pointed at it right now. Around
Okay.
260, something like that, a little
Yeah.
bit more.
Yeah, I was thinking 260. A little bit south of west, because that's where globally we need to
Look, a
head.
sponge.
Yeah.
And maybe a coral.
Okay, so should we start off with a 30 meter?
Yeah,
Yeah, I'd be comfortable with that.
that's fine with me. At, what's the heading?
260
Copy that. Sounds good.
at 0.2 knots. Bridge, Nav. Bridge, could I have a 30 meter move?
So, shore-based
Bearing
scientists.
260.
I know the feed may not be
Speed
fantastic today.
0.2 knots.
How would you like to accomplish our objectives today? Do we want to try to zoom in on things
Good
for a
copy,
while
Rich.
that we haven't seen yet? You know, when we first start and check out the sponges and the corals and all the other interesting stuff? I think that would be me and Diva's preference.
Yeah,
Thank you, Bridge.
my vote would be we're a little more selective. Here's a couple corals that would be worth zooming in on.
Okay.
Yeah, so
Get a
pilot,
few
can we
things
zoom in
and
on a
try
couple
to
of
get
these
up
corals,
on the
please?
Summit, because that's probably where we're going to see a lot more stuff.
Okay.
So this looks like a Christogorgid.
That was what I was going to guess.
it's because they're the really bushy ones.
Yes, exactly.
Yeah. So Christogorgid with a squat lobster commensal inside of it. Just below there's a comatulate crinoid and what looks like an icidid bamboo coral just below that. Oh no, actually perhaps that's a primnoid.
Do we want to go down there and check it out?
Oh, I'm thinking maybe primnoid now.
I don't know.
No, icidid. Oh, I don't know. Chris, I default to you.
Better shot. Stand by.
I see why you thought it was a primnoid. If I had to guess right now, I'd say it's a primnoid too.
Guess so.
Yeah, it looks a little Calyptrophora-esque.
Okay, you want to come in?
All right, coming in.
Mm.
I'm thinking primnoid. Any scientists have shown no anymore?
I'm guessing it might be a Candidella. It's got 3 polyps per whorl, and the polyps are pretty big. I don't think it's Gigantea because it looked like it branched at the base somewhere. It doesn't look right for that, but definitely a primnoid.
Okay.
Okay, you want to
And pilots, if we could zoom back out, and then there was one more colony further up, which we hadn't gotten to
Holding
see
partial.
yet.
Copy.
Or to the right. Yeah, let's do the one to the right,
Copy.
please.
Okay, Brian.
What's
come
the
on in.
little thing
Coming in.
over in the right-hand corner that just went off frame?
Oh, I didn't get to see it.
All right, that's full.
Copy.
So this is another one of the primnoid corals, and this one's got an Ophiocanthid brittle star living on it commensally. And pilot, one more thing. So off to the right, there was a gelatinous-looking animal
Coming out partial.
right there. I think it's a sponge.
Sponge.
And there's a barnacle above it.
Yeah. You can comment on that if we want.
Coming in.
I'm gonna bring my sonar in just
That's
a little
full
bit.
zoom.
Yeah.
Copy. Teetering here.
So the last primnoid was likely Norella macrocalyx. And
Look at those sponge spicules. Is that what they're still called when they stick to the rock?
yes, yes it is.
We're done with this little guy?
Okay, yeah, no, that's great. Thank you, pilots.
come along.
Poliopogon?
So
How do you
we're
pronounce it?
Poliopogon.
Poliopogon.
What's that?
I'm
Thank
never
you,
going to get
Chris.
this right.
So that's a
again?
baby
Where do you start working on
Poliopogon.
that?
We saw a couple much larger individuals on a couple of
20
the earlier
meters out.
dives.
We did a 30-meter move.
That's right,
Okay.
30 meter.
Okay.
We've got about 10 meters left in it, a little bit less.
Looks like there's a big sponge up here.
Yeah.
Patty, aren't you excited?
You
We're
should
looking
be
at
moving
rocks.
now, co-pilot.
Real rocks.
Copy that.
Well, not
Just
only am I
started
excited, I
maybe
think
a
I
minute
just
ago.
saw a dike.
At most.
You think so? I could believe that.
Yeah, I saw some cooling cracks.
Awesome.
Little ones. I'm just wondering if this whole thing is relatively, can't be done.
It could be. I mean, these are definitely blocky and
It's a big sponge.
quite big. Oh, look at those sponges. It's like a little convention of sponges.
Convention. Okay. So here, doesn't sound like I'm on. Am I on?
You are on.
Okay. So here we've got a couple more sponges. One of them you can see has got a commensal shrimp, I think, by the looks of it.
Do we
And these are
snap
more
on that, Brian?
polyopogons,
Thanks.
I'm pretty close.
but
to
just
full
much
partial.
larger.
Okay.
You have a visual on this?
Yeah, let's keep you coming up.
Okay.
Let
Yeah,
me go up
beautiful
if we need to.
Copy that.
shrimp inside of this polyopogon.
Calling in.
And so this shrimp could be a type of labaeid,
Just
rather
gonna leave it
from
here,
the
Brian.
genus
That's
Labaeus.
Copy that.
Okay, I'm more comfortable.
And they
now.
tend to
My sonar
inhabit
can see
sponges
25
commensally.
clear, so
Yeah, that sounds good.
Great, thank you, pilots.
no problem.
Just hold that zoom.
I'm comfortable here.
Copy.
So that ship move is done, pilots.
Okay. So I'll probably swing for another couple minutes.
Yeah. Maybe another 3.
Okay.
Any associates here? Well, they don't sound too excited, Brian. You can come wide.
Coming wide. Full
So
wide.
should we go ahead and put another one in? Watch leads have asked us to.
Yeah, go ahead.
All right. Looks like the heading is still pretty good. Co-pilot.
Yeah. Yep, I'm happy with 260.
I'll go out ahead.
All right. I'll call in another one then. Bridge, nav. Bridge, could I have a 30 meter move? Bearing 260. Speed 0.2 knots. Good copy, Bridge.
Some big rocks.
Pilot, aren't the rocks nice today? Don't
Yeah.
they look great?
They're nice every day.
Good call.
These are good.
Good answer.
Is that the right answer?
So
That
as
was
we
totally the right answer. Sorry, I was
I
having
feel
a
like there's
moment.
some pandering going on.
All right, so we are on the hunt for corals and sponges and exciting biology.
And we can see quite a couple isolated whip corals as we zoom over this rock feature.
It's all tilted.
You can lose them, Brian. Thanks.
Actually, there's a lot less sediment than I would have expected for this area. Right now, at least.
Yeah, Deb, we were just talking about that bodes well for the summit maybe being relatively sediment clear, at least in our opinion.
Hope so. That would be great. But now I'll put Diva on the spot and have her explain why it would be better if we had bare rock rather than a sedimented flat top.
For what?
Why would it be better?
For
to have-
biology?
For biology, yeah.
Because it means they can anchor more easily.
Thank you, Diva.
But with that said, we're still not seeing that much biology here.
True, not yet.
That could be because the currents are too strong here, or there aren't enough larvae recruiting in the area,
Or we're just not to the top yet.
or there's not enough food. Lots of different things.
Push.
Water chemistry. But, ooh, what's in this crevice?
Sediment.
Straight ahead. No, there's definitely an animal. Oh, that looks like one our pilots know.
I know this one.
That
Do you
looks
know
like
this
a
one now
Ritagorgia
too?
Right there.
Iridogorgia.
Very good, everybody.
I feel like I'm in like a biology class.
This is when they write on the chat log, that's
It
not a
doesn't-
Ritagorgia.
It doesn't look like that though, because there's different like.
Is it Iridogorgia?
It looks like,
I'm
I
not
was
sure it is.
thinking it was like a spiral trapped in the crevice, but now...
That was a misleading question. Come on, NVIDIA.
No, they're not Iridogorgias.
You led us astray.
I did.
So what are they?
It's a setup.
Plurigorgias. Plurigorgia militaris,
There's two different kinds, aren't there?
which is a
Brian?
type of chrysogorgid.
You only knew that because Chris put it in the event
Yep.
log.
Keep coming.
Yep. But did you see where I was going? It did look like a spiral in there.
In the background there, isn't there? Step by one.
Oh, and so this one's got a
See back
couple
there.
hydroids growing on it.
Okay. The clear. Okay. Copy.
But what would make these corals choose to live in the cracks of a rock and not out in the free-flowing current?
So I think that's more of a Chris question, but I can hypothesize, throw a hypothesis out there and say that perhaps this is where they're more sheltered from currents in the area, and so it's sort of a safe refuge where larvae that may have settled on the exposed faces of
All right,
rocks
Theo's
were washed away
Come on.
or predated upon or something,
Trying to
but
take off.
these ones in this crevice were more sheltered and therefore were able to grow into adults. But Scott or Chris can probably answer that better than I can. Ooh, and here's an anemone. We haven't seen any anemones on substrate, on rock substrate for a while. They've mostly been attached to other animals.
This is a great picture. Beautiful geology with a very nice anemone specimen right in the middle.
It's like an accessory, is that what you're saying?
It's totally an accessory.
You looking down at me?
No, I'm only at 37,
Yeah,
so yeah, we're happy. That's 45.
Copy.
ACM.
Big delta.
So this one might be an Exocoelectus,
Want to come in here, Brian?
if I can get a closer look.
Or-
Yeah,
Holding.
I'm pretty happy with that ID. You can see the very characteristic little baubles on the ends of the tentacles. The tentacles are also quite small in comparison
Coming
to
wide.
the column.
Come on. It's auto X1.
And actually, in all seriousness, it's actually a good look at the rock
Go
as
back
well.
in.
And what you can see is this, what looks like a
Ooh.
very heavy manganese crust. You can see that rounded shape that is characteristic of these manganese crusts. So all kidding aside, this was actually a good zoom for geology as well.
Try to come up and we
Good.
can look down.
That's
into
what
it.
we like to see.
Okay, pilots, we made that next ship move. It's complete.
Copy that.
So,
Before we call the next one in, I'd like to push out a little bit. Just because that
Yeah, let's
sonar
wait.
return.
Let's wait 1 second. Okay, Brian, I think they're down here anyway.
All right, coming wide.
Copy. Let's get out.
You're full wide.
You're back on. Okay, look.
There are a lot of these unbranched corals.
Okay, so yesterday we investigated a geo similar to this one, and at the beginning of the dive we touched down in an area where there was a lot more coral and sponges, and this sort of increased as we went up the ridge, and also changed slightly in composition, where we began in an area
come
with
up.
more isidids and primnoids
Come
and
up.
ended in an area with
Yeah, both
a
of
lot
us.
of iridogorgids, which are iridogorgias, which are a type of chrysogorgid. But here we're seeing much less coral. There are lots of dead coral stalks around, but
Out there.
much less of the live, large colonies we saw yesterday. This could just be
We're kind of look
you
down
know,
this
this
crack there,
area,
Brian, as
and
we
we
come up
should-
and over.
count our chickens before they hatch. But
You can go partial.
pilot, what's the
Hi
current
there.
like here?
It's hard to tell. Depends on how close I am to the wall. It's really not bad at all either way.
Okay. Yeah, so we seem
Yeah,
to
it'd be
see.
good to see what these corals are in the crack there. Unfortunately, our screen's pretty messed up, but we're hoping to get a glimpse.
So there's definitely some chrysogorgids along this
Fracture.
along this
so how
fracture,
about now? It seems like
as
he's
well
pretty
as some
far
of
ahead.
the
I mean, you got 45 degrees before you're on top of him.
Plurigorgia militaris.
Let's give it 1 more minute. Which one do we want to look at first, Watchley?
Let's start in further inside the crack. So the ones further down, please.
Okay. Come on in, Brian. Down there.
That's all I got.
Was that a sponge too?
I-
Outside?
Sliding on you.
Well, now I've said it is.
Yeah, so these look pretty similar to what we were looking at before. There's definitely some Pleurogorgia as well as some Chrysogorgias. And you can see a couple of them have these
Come on, Brian.
beautiful commensal ophiuroids living on them.
Take my tilt.
Bull wide.
And can we, look at this bushy Chrysogorgia,
Catch
please?
tilt.
And they look like there's some more Poliopogon sponges around here.
Okay.
Video, let me know if I gotta go up or down.
Go ahead in.
Right. Going in on Chrysogorgia. Partial.
Okay, you can come in if you want.
Right, going
however.
full. Setting focus. Pulling out a little bit.
Give it a tilt down there.
There we go.
Nice shot.
Is that maximum zoom?
No, I can come in a little bit more. You ready, pilot?
Go ahead.
There's Max.
Beautiful.
Can you tilt me up a little bit, co-pilot?
Yeah.
Thank you.
Okay. Light's a little better.
Perfect there.
We got about 30 seconds here.
All right, video's clear. Ready?
Yep.
All right, coming wide.
Go ahead, tilt back.
So this,
Go ahead. Yep.
that
Thank
little
you.
squat lobster we could see inside of the Chrysogorgid was likely from the genus Uropticus, which is a type of chirostylid squat lobster, and these are typically seen inhabiting these
Okay.
chirostylids,
Get that
usually
in
the same
now, Andy.
species of
Thank you.
chirostylid.
Copy that, pilot.
So it's really interesting. If you look at the Serios
So the heading
feed
still
on
looks
stream
good to
2,
me at
you can see the
260.
sponges because they're so light.
I agree.
They just sort of
Okay.
stick out everywhere. It's really quite fascinating. See?
Oh yeah.
Bridge, Nav. Bridge guy with 30 meter move, bearing 260, speed 0.2 knots. Good copy, Bridge.
We can make a decision.
Oh,
Wow.
so do we think the sponge is dead?
What is that? Do you think that's a sponge?
Thank you, Bridge.
I mean, what else
ahead,
could
Brian.
it be? It
I don't
must
know.
be a sponge.
So it's a dead sponge, but it's a pretty curious one.
So interesting looking.
Chris, have you ever seen
Kill
anything
wave.
like this before?
I have not, but unfortunately, it doesn't make a good collection because dead sponges lose crucial microscopic skeletal parts, the spicules, that we need to look at to identify them.
So our other sponge expert, Charlie Pomponi, says
Pretty
it's
comfortable.
possible it's not dead and it
What's
may
that?
be
I'm pretty
a,
comfortable.
a lithisted instead. I
can
love
see out.
lively debate among
Lithisted.
the sponge people.
Okay, on center. Yeah, good to know.
Do we want to zoom on something else?
Yeah, let's see what else is in the area. Where
Coming with.
Oh,
are we
on,
on
yeah.
the ship move? Are we?
look at these giant sponges. I think they just put in another ship.
Can we have the lasers on just for a second, just so we could gauge how big they are, please? Okay, so each one of those sponges is about
Come back on.
40 centimeters
Ah,
long.
There you go.
I got you at 80 degrees.
We got lasers on the dead one when we first came out to it.
oh, good.
And they're really white.
Yeah, it's tricky. I think those
What is
things
that?
reflect so much.
Is that dead sponge?
Get some lasers on this
That
guy.
looks like the base of a sponge.
It's a big sponge.
That's either that base of that sponge or the base of another sponge which has died. Perhaps.
These are very big sponges.
What's that? Is that a dead stalk up there?
Can we
Yeah.
zoom in and
Yeah.
check?
That's what
It looks like it.
You can go snap zoom.
Snap zoom eye. Coming in.
Something on it.
Yeah.
But with an ophiuroid living on it and some hydroids.
Well, as we saw yesterday, we saw a lot of stocks that were
Okay. You can
a
go back
great
on up.
place to set up your community.
All right.
Full wide.
Just carry on here.
Coming up. Big heaves.
Yeah.
Can we zoom on this pink coral, please? We haven't seen one of these for this dive yet.
Sure. Stand by video. Okay, come on in.
Right on the end?
Yep. Whoever that was,
Yeah,
we didn't
so
hear you.
this
Oh,
actually is a species of corallity. It's the same family as the precious corals, and it's probably hemicorallium.
Great, thank you, Chris.
Little space.
Big base in that one.
Yeah.
it's got some
And
commensals.
it's got some, yeah, commensal ophiuroids living on it.
Let me know if you want to do it tight anywhere, Brian.
I think I'm just about full zoom. I probably got just a touch left, so I think we're
Okay.
good.
Copy.
Video's ready when you are.
Okay, you can come on.
Full wide.
Copy. Anything else on his face here, Watchley?
Crinoid.
Yeah, let's get a look at that crinoid, please.
Stop.
Yeah, please. I was wondering whether that's comatulate or whether it's stocked.
Definitely stalked.
Stock written all over it. Come on in, video.
Holding partial.
Copy.
So,
Stock,
ooh,
right?
no, I don't know. Can we zoom a little bit more at the mouth area, please? Just so we can
Mouth.
try and see the
It might-
whether it's attached to the stalk or I think it is stalked.
I mean, it seems to look that way. It doesn't look like it's living
There
on
is a
something.
lot of growth on the stalk though, which is what's concerning me. But
see what
no,
you're
that
saying.
might be
Okay,
normal.
Come on in, video.
Coming in.
No, that's
That's all
definitely
I got.
stalked.
Copy. I'm not going to get any closer. It's stock.
Got you at about 80 degrees.
Yeah.
So
When you're done, video, let's come wide.
Coming wide.
Oh, what's that?
So that might be a Bathycrinidae.
Copy,
stalked.
thanks, video.
Yeah. So Chris and I came to the same conclusion. Bathycrinidae. I agree with you, Chris.
What is on the Sirius
Okay,
feed right
pilots,
now?
that ship move's
I'm
done
trying
as
to
well.
decide if it's just shadow. It's weird looking.
What's that?
I think it
That
is
ship
a shadow.
move is done.
Okay.
It looks like the local high is a little bit to our south now, maybe 245, something like that.
That would make sense with the high pack, right?
Yeah.
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 That sounds good if you want to get that one in here in a minute. Let me push
These
out.
rocks are
Sure.
so much more
Sure.
intact than yesterday.
2, 4, 5.
Yeah, and they're also less rounded. I mean, from
Yeah,
a
no, you're totally right.
yeah.
I mean, they definitely still have that manganese crust coating because when we zoom in on it, we can really see it. But you're totally right. Yesterday, everything was
What is that,
very,
Brian?
very rounded, and it was mostly very low
For sure.
relief. Remember that? You know, it was pretty much close to the ground. These are big blocks of rock.
Can I go snap? Hold there. Sponge.
So another
Perfect
sponge
sponge.
down in this
Maybe
crevice.
a little bit more.
Could we
co-pilot,
zoom in
maybe
on this one?
240.
On the dead sponge.
This one looks
in
like
the
a
crevice?
polyopogon,
looks like
but
it's to my left
It's
a
not
little bit.
dead, is it? I thought it was
that-
alive.
I think in the at the forefront, there's some dead tissue,
charge
Right.
which I think
240
maybe
was
from
a
something
lot.
else,
Yeah.
Copy.
and then the
Kitty right there, I
sponges,
think would be fine.
or the edge of it is dead.
Yeah.
You can could zoom a little more video if you got it.
That's it.
Okay.
Great. Pilots,
Great,
I'm going to
thank
call
you,
in
pilots.
240 then.
And there was also a Primnoid in the foreground.
Sound good?
Yeah, I'm lined up.
Do we want to look at it?
The Primnoid?
Partial.
With the sonar?
Not really.
Yeah.
Okay.
All right, coming full.
Would you?
Full wide.
It's probably the same one we've seen already.
Bridge, nav.
240 pilot.
Copy.
I'd like to request a 30 meter move. Bearing 240. Speed 0.2 knots. Good copy, Bridge.
Yeah, let's come up here. That's a overhang there.
Look at that face, how sheer.
You want me to turn to keep you in view, or
No, come back.
Okay.
Just doing a little pirouette around the end here.
I'll just keep my delta right at 15 with you.
Looks like a little hole
Copy.
boy just left of the lasers.
Copy.
Can,
Fish.
Fish.
oh, so can we zoom on the fish, please?
Sure. You can turn to look at me. Okay.
Those almost look like pillows at the top.
And Deb, I think those are pillows.
That's so funny. I was just thinking the same thing. In fact, I think I
Okay,
just
come
said
on
the
in
same
video.
thing.
You did.
I'm going to translate to the right. as
Patty,
we
great minds
shoot
think
now.
alike.
So this is an ophidiid, which is a cuskeel. The very round head makes me think it's a Basozetus, and yes, Bruce Mundy has just said the same thing. And if we were to get a shot of its eyes, they'd probably be quite small and beady.
It's like a pretty hard sonar
Creeping in
return
on
for
it.
me.
Yep,
Keep coming in.
small and beady.
Keep coming in.
Hold there.
Holding.
Oh my gosh, stop turning.
You talking about right in front of you, co-pilot?
Yeah.
They almost look like tadpoles, you know, they're kind of
There wouldn't
just
be any
out of
more
proportion.
returns behind it
Hold
if it was
there,
a hard
video.
one.
See if we
Holding.
can,
Okay.
Coming in on it. You're getting closer.
Yeah.
And
Just
if you're
be a
following
bright
on shore, we
line
do
with shadow
appreciate
behind it.
appreciate you staying with us despite the
Yeah, I'm just seeing
temperamental video feed.
how I don't see anything right here.
Yeah.
So
Come a little wide.
Coming out a little.
I want to get his eyes, but he keeps rotating the
Full
opposite
frame.
way.
it's a trick.
He's not interested. He's like, don't look at me. Don't look at my beady eyes.
Nice maneuvering, pilot.
Okay. He's still rotating. He's in pretty tight.
Okay,
Yeah, he's
that's great,
inside the
pilots.
rifle.
Thank you.
Coming wide.
Stop antagonizing the fish.
Full wide.
Copy.
Nice flying.
He's playing games with you.
I know, I think he is.
So
Alright,
can we
let's
get
see
a
if we
zoom on that really big sponge dead ahead, please?
How far did you come over?
I'm 220. Move is
240.
240.
How far in?
Oh, look,
Whatever.
there's a lot more stuff here.
About halfway.
Maybe not a lot.
This is off our track. We're going to zoom from here, though.
Oh, no, that's okay.
So don't zoom.
If you can do a snap zoom,
Go ahead, video.
we appreciate
Up.
anything. So I'm not sure what kind of sponge this is. Maybe Chris or Shirley will know. But it has a quite large feather star or comatulid crinoid living on it.
Yeah, pilot.
Yeah, so we're seeing the backside of a sponge called Tritopleura. It would probably be,
Full wide.
you probably recognize
From
it if you saw the
yesterday.
other side of it. And these are fairly common at these depths. They're really interesting, and
give it
right
1
now
sec.
we only know them to genus because I don't think any of the species have been described that at least we collected in Hawaii.
Copy.
Yeah, so Chris, those are the ones we were referring to yesterday as the leaf sponge, correct? I think so.
30.
All right, so we are here at Enrique Seamount. We are at a depth of 2,210.
A little
I
bit.
will just remind everyone that today we are having some difficulty with our video feeds. So it's very helpful for the shore-based scientists to please let us know in the event log if you're having any luck with I-1 or I-2. But for those of you who are following along and for our shore-based scientists, we apologize very much for the lack of good video, and we hope you will stick with us for the next couple of days. Hopefully it should get fixed.
Biological life. Southbound to here.
South, yeah.
And on that
Since we're
note,
getting closer to the top here.
that move is complete as well. So,
Okay.
So, Chris,
Get
we're
another
really
one
not
in.
seeing the same density of
as
corals
far as
and
local
sponges
high
that we
goes,
saw
doesn't
yesterday.
really look
Is
like
that
we have a
correct?
clear return. I think maybe we should cut even farther south.
Yeah, not so far. Yesterday, as I recall, we kind of dropped right on the summit area, I think, or pretty close to it. So I suspect we're going to see some more things as we go up. And if you look at the top of the screen, there look like a couple big isitids, the branching ones that just showed up for the first time.
We can stay along this one. Or
Pilot,
232.
are there corals at the top of the screen right now?
There are.
Yeah, they went off to the upper left. They're now off screen.
Would it be possible to zoom in on those?
These ones.
Thank you,
way
Bridge.
out there?
Yes.
Yeah, we're headed that way. We
Okay,
can't
perfect.
get there now.
All right.
But,
We'll stare at them from afar, trying to figure out what they are. Chris, we're moving in that direction right now.
So I'd say somewhere between 230 and 220. We're on 220 already.
I like 220.
All right.
That's right where the biological that they're looking at is, isn't it?
yeah.
All right. I'll put a 30-meter move in at 220.
Sounds good. What have we got center screen out there, Brian? The translucent.
Holothurian.
Okay. Is that
Bridge,
another
Nav.
stalk rhinoid in the foreground?
It is.
Bridge, can I have a 30-meter
a
move
holothurian in the middle of the
bearing
screen.
220 degrees?
It's like there's an echo.
Speed
Pilot, do you see a
0.2 knots.
holothurian anywhere?
We do.
Can you zoom in on it?
Good copy, Bridge.
Sort of a bluish.
I got a pretty big delta.
Yeah, so it's smack in the center of the screen, just above the lasers.
Yeah. We,
They're getting there.
There also
They're a little
looks
far away,
like there's
I
a
think.
cauterized sponge next to the crinoid.
to the crinoid. Oh, yeah. You want to go snap on that?
Sure. Oh, I was wrong. It's a black coral.
Pathopathies?
Probably not.
Darn it.
So I think our pilots may actually be right again with that ID. That does look like an
So this
Antipatherian.
is a black coral.
Yeah, a black coral, known as Bathypathes.
Nailed it.
That coral, though, and the one on its right, they don't really look like they're in very good condition. You can see all their skeletons been
Like a
exposed
Holothurian.
and overgrown.
Yeah.
All right, coming wide.
Great, thank you. As well as there's a stalked crinoid in the middle. So yeah, that bluey. I think it's just a sponge, though. Not just a sponge. Okay. Oh no, maybe it is a holothurian.
As we zoom in on it, you can really see that
Gut.
rounded manganese crust texture that we've been seeing in
Oh,
so
I thought you
many
were talking
places.
about the holothurian.
No. That's why I said manganese crust. The holothurian does not actually have a manganese crust.
Come on in.
Not yet, anyway.
Soon everything has a manganese crust.
So this is a holothurian.
Oh, no. I can feel
Perhaps most closely related to Hansenothuria, which is a type of synallactid holothurian. And this one hasn't had too much to eat
Yeah.
yet.
Want to zoom any particular place?
Yeah, it is.
No, I'm fine.
Okay, great. Thank you, pilots.
Coming wide. Full wide.
Copy.
Losing the skids.
Thanks. There's a shrimp about to photobomb us all here.
Oh, and a shrimp's just come dive bombing.
It's always
our
nice when that
image.
happens.
Yeah, photobomb. And that ophiuroid just made a leap for it a little while ago.
I always miss that. What's in the upper right over there?
A sponge.
Okay.
As well as around here, we can see some stalks that have been covered in manganese crust, some live isidids, quite a bit of sediment down in the cracks.
We're going to come take a look where Chris Kelly was looking earlier. Over here.
So yeah, I think they want to get a closer look at some of these isidids.
Okay.
Please.
Do we think all of them are the same, or you want one in particular? Okay. We will pick this one.
Who? Oh, pilot, sorry, you asked me a question.
That's okay.
Yeah, just take initiative.
We get yelled at sometimes when we do that. Come on in, video.
Pilot, we're deeply sorry for that comment.
He knows I'm
Hold
joking.
impartial.
There's at least one sponge back there as well.
Yeah, a bolosoma, like what we collected yesterday, maybe. So here are some of these isidid corals. Is that the most zoom we have, video?
Go ahead, Brian.
Negative.
We just want to see if this one had the same veining as we saw towards the end of the dive yesterday, which was very strange. Does it?
I'm losing you just a little off to my port there.
Copy.
No, I'm not
I
convinced.
don't know. I'm not
no, I don't
convinced
think
either.
it does.
I'm at 56 degrees. though.
Scott?
So
Hi, Diva.
Scott,
Hi, Deb. It's Scott here.
hey,
Come way
we were
up,
just calling your
look
name.
back at me.
Yeah. Hang on, I have to lean over and mute this. So as you know, the video is a little bit pixelated, so I can't tell if I'm seeing bumps or if that's just pixelation on the tissue between, but it looks like bumps, huh?
It does look like bumps.
I mean,
There's
no,
a few.
So I think, looking at it now, Chris, I don't think this looks, not Chris, Scott, I don't think this looks like what we saw yesterday.
No, but that's not what he's saying. He just sees some.
Oh,
Are there
yeah.
bumps
So there are
on
definitely
the-
bumps, but
Yeah.
it doesn't look like the actual
There are definitely
veining.
bumps. It
Right.
isn't just the pixelation.
Right.
Okay,
No, that's what he was asking.
Um,
Yeah.
On the ship, you're a bit hard to hear, but I heard that from Chris. We're breaking up a little bit. The reason I said something about the bumps is last year on our fall expedition to the Pacific Remote Islands National Marine Monument, Marine National Monument, near Johnson Atoll, we found a bamboo coral that had all these little white bumps running along the tissue between the polyps, and we guessed that those might be what are called nematocyst batteries. Basically, they're little packets filled with these stinging cells, and it's something that you would see on as a defensive strategy.
Okay, let's
And
get 30 more
in that
seconds and then I gotta
veining
run.
pattern that we saw yesterday, I wondered if that could be the beginning of the
20
development of something
zoom
like those
out just a
bumps,
touch to
because
get a better
the bumps were kind of oriented in the
establishing
same way
shot.
as the
Kill
as that
lasers.
pattern of veining that we saw. And we won't know until the samples are returned to shore in a couple of months and we get to look at it more closely. But if this has bumps, it
All right,
could
video's
be
good.
related
Copy.
to that
Coming along.
sort of veining.
Copy.
So this is all interesting. It's really great to see these sort of repeated patterns, and with the couple of samples that we're lucky enough to get, we'll check that out. So maybe the next time we see this in coming years, we'll be able to say a little bit more about it.
So, Scott, what
Yeah,
would it
looks
be
like the
using
local
the
highs around
nematocyst
230
batteries for?
now.
For prey? Or for
And
protection?
that ship
What a
moves complete,
fantastic
so once you
question,
guys
Diva.
get sorted,
Okay.
No,
Yeah,
I
we'll
think
try
it would
that
be more
trench
as a
out.
defensive thing. And
Right.
there are lots of different invertebrates that will feed on bamboo corals, and it's quite interesting because last year we saw
Yeah.
many instances of predators on bamboo corals, very commonly sea stars, asteroids, but we also saw there's a special kind of molluscan related to snails called an aplacophorn. It looks like a little worm, but it's actually a mollusk, and they're also specialist cnidarian predators,
Yeah, sure is.
and we would see those as well. And so those are both very small, and they're essentially feeding at the level of polyp. I guess the starfish is bigger, but perhaps it's related to that. I really don't know. And
the reason I can say we don't know is when we saw that last year, it's the first time we ever saw anything like that.
So then last question, Scott. Does that mean that if we had collected a piece of this, I should be careful taking it out of the bio box?
A whole bunch of
We want you to be quiet.
parallel pillow rules coming down the side of this slope.
I agree, Patty. We cut off the biologists because we need to talk about the geology for a moment. So this is always great. You know, we
Wow,
really,
look at
really
that
love
relief.
pillow basalts. And if you look in the Sirius camera right now, you can really see it was just a huge wall of pillow basalts all sloping in the same direction. It's fantastic. What's a day without a nice pillow basalt?
It's a sharp ridge, right?
Yeah.
That's cool.